Hello, my name is Christian Kiss. I'm an instrument specialist with ThermoFisher, and today I'm here to talk about some of our products used for automating workflow. In particular, ThermoFisher sells a wide variety of products, among them instrumentation for making the processing of various different techniques a lot easier, faster, or even more accurate. Among the things in this poster here are things like the multi-drop combi used for automating plate filling, a versette used for automated liquid handling for pipetting and dispensing different volumes of liquid, the Kingfisher Flex for DNA and RNA extraction, the Multi-Scan Go, which is a multi-plate reader for doing UV-Vis type readings for absorbance, and then of course the PicoReal QPCR instrument for doing real-time PCR analysis. To demonstrate that we can string these instruments together for a workflow and to make things a lot easier, we put together a particular process demonstrated in this poster. First of all, we started out with nucleic acid purification of blood samples. Basically, this was put into the Kingfisher instrument. In order to facilitate this kind of process, of course, we want to automate liquid handling as well because we don't want to sit there and pipette all of our reagents. So the multi-drop combi was used to put all of our wash buffers into the plates. In Table 1, we have a number of plates, essentially the inventory of things that are going to be put onto the instrument. You can see that they consist of a variety of buffers, and wash buffers in particular, and dilution buffers. The volumes are listed, but the multi-drop combi was used to dispense all of these reagents, so none of this has to be done by hand. From here, once the DNA extraction was done on the Kingfisher instrument, which can automate that particular process, and you can get all of that done in about 25 to 40 minutes, depending on the type of material that you're working with, we want to move on to evaluating the material that we collected. And so one of the things we want to do is take an absorbance reading to see what kind of concentration or yield that we got from the process. In order to do this, of course, we want to dilute out some of the sample material that we got from the Kingfisher. And so a 1:4 dilution was made of this. This was dispensed using the versette liquid handler that we talked about, essentially taking 10 microliters of our DNA sample and then 30 microliters of water and just mixing that together. Once we did this with the liquid handler, the plate was put onto the Multi-Scan Go. This takes a UV absorbance reading. We took a UV 260, 280 measurement and a 260, 230 and some background readings, of course, for normalization. Once we got the readings, we were able to determine concentration of the DNA, and we can carry it forward into additional assays. The assay that we're going to do is going to be a QPCR assay. Normally for this, what you want to do is adjust the concentrations so that they're equal to each other. We can also do this with the multi-drop combi, essentially just program in the amount of volume that we need to add to each of the wells to normalize all of the templates down to about 10 nanograms per microliter. Once we were able to do this, this went into real-time QPCR analysis. There were two different types of QPCR analysis done. For CyberGreen, we looked at the chromosome Y detection to determine whether it was a male or female sample, and then probe-based analysis to look at the RH factor. This was put onto the PicoReal instrument, and the results are shown over here. Basically, your green lines are male determination, so when you have a curve for the green line, you have the presence of a male. Flat green lines denote the presence, the absence of a signal, so basically the presence of a female. The red lines are for the RH factor. You can see in this graph that the red line gives you a positive. In the second graph next to it, the red lines are relatively baselines, so there is no RH factor or RH negative. All of this can be strung together to automate the entire workflow, basically making things a lot easier and a lot faster, and in many cases making your data more precise because you're taking the human element out of the entire process. Thank you for your attention, and for more information, please contact us at thermofisher.com.